So welcome to the first lecture on cells. This class is going to cover the cells, and this beginning portion is the chemistry of life. I'm Wendy Nofke, one of your instructors that's going to be presenting this lecture. You'll have other ones in the future with Lauren and myself. So all living things are made up of chemicals, and so we need a little bit of background on chemistry to get started. Matter is anything that has mass and occupies space. So anything you see around you that you're able to see visually with your eyes, anything you're able to touch, is going to fall into the category of matter. Elements are the different types of matter, or the pure substance. Down at the bottom of the screen you can see an example of what a periodic table looks like, and in each of those squares you see some letters, and those letters are the chemical symbols for the different types of elements. In chemistry, a compound is going to be made up of two or more elements. A molecule is going to be made up of two or more atoms of the same element. So an example of a compound would be something like carbon dioxide that has one carbon and two oxygens. An example of a molecule would be something like oxygen gas that's made up of two oxygens. Compounds are going to be held together by chemical bonds, which are going to force holding the molecules together. A chemical reaction is going to be the process of converting one of the chemical substances into another. When we have chemical reactions, on one side of the equation we have the reactants, which are your starting materials. On the other side you have the products that are the ending materials. You can think of this like baking something in your kitchen. The left, your reactants, are your different ingredients, and the product is what you end up with after you pull it out of the oven. In chemistry, we divide it into two main branches of study, organic and inorganic chemistry. Organic chemistry is the chemistry of carbon, or the study of hydrocarbons. It's looking at carbon with the four bonds that it can form. Down at the bottom, the little graphic shows the carbon molecule in the center, and that it's got four molecules that it attaches to. Inorganic chemistry is going to be the study of all the different elements. This will include carbon, but carbon won't be the star of the show in inorganic chemistry. In life, we have four major macromolecules that are going to make up all of the substances that we find in different organisms. The first one is the carbohydrates. Some examples are sugars and starches. This is what living cells use to provide energy and also make some of their structures. Your lipids are your fats or oils. Cells use these to make your energy stores and also create membranes. Proteins are going to be used to make things like enzymes, antibodies, and structures. They will also help to carry messages in the cell. And then your nucleic acids, these are going to be the DNA and the RNA. These are your genetic instructions for everything in the cell. So looking at the carbohydrates, you'll see the terms monosaccharides, disaccharides, and polysaccharides. A saccharide is just another term for a sugar, mono meaning one, di meaning two, and poly meaning many. So a monosaccharide is going to be just a single sugar unit. Common examples are glucose, fructose, and galactose. Disaccharides are two of these sugar units attached together. Lactose is composed of glucose and galactose. This is your common milk sugar. Sucrose, which is your common table sugar, is glucose and fructose together. And maltose is a malt sugar, and that's glucose and glucose attached together. With the polysaccharides, we have a few different forms. Starch that you'll find in plants, glycogen in animals. Cellulose is also found in plants but is more structural, and then chitin that you'll find in insects. The lipids, which include your fats and oils, are going to be long chains of carbon attached to many hydrogens. When they're saturated, the carbon is going to be bonded to as many hydrogens as possible. This is going to be a very stable form for the carbon to be in. As you can see down below in the graphic, there is an example of a saturated fat here, and each carbon is attached to hydrogens on at least 3 of the legs, some of them only on 2 of the legs because they're attached to neighboring carbons. When you have an unsaturated fat, some of the carbons are not going to be bound to the maximum number of hydrogens. Instead, you'll have a double bond where it will be bound to a neighboring group of carbons. The lower picture in the graphic is going to show you an unsaturated bond where you can see a carbon and a double bond attached to a neighboring carbon. You can also see that it bends the molecule, causing it to be slightly less stable. When you look at saturated fats, they will tend to be more stable and more solid at room temperature. Your unsaturated fats are less stable and they're going to be more liquid at room temperature. With the types of lipids, the most common one is the triglyceride. This is the main form of stored fat in animals. Phospholipids are going to have a phosphate head on them. These are going to be used in cell membranes. And then the sterols are going to be ring-shaped fats that are used as chemical messengers. They're used to make hormones, and the most common form of this is cholesterol. Proteins are going to be used to make structures in cells. They're made from 20 different amino acids in various combinations. These are found in all living things. Below you can see an example of what the amino acid structure looks like. In the center you've got a carbon atom, and on one side of the carbon atom it's attached to a hydrogen. On the other side it's got a carbon attached to two oxygens, and then an oxygen and a hydrogen for an acid group. Over to the left it's attached to an amine group, and then on the top you have the R, that is the variable group. In all 20 different amino acids, the only thing that is different in the structure is what is located at R. Polypeptides are just long chains of amino acids, and they will fold up to make different shapes. The picture to the right is an example of a polypeptide that is folded up into a protein. When proteins form their structure, there's 4 levels to the structure. The primary structure is just the sequence of the amino acids, and that's going to form a nice chain of different amino acids. The secondary structure is going to be when those initial chains of amino acids fold up into alpha helices or beta pleated sheets. This is due to hydrogen bonding in the neighboring amino acids. The tertiary structure is some additional folding that you're going to have upon the secondary structure. So you will still have the alpha helices or beta pleated sheets, but they do some additional folds. The final structure is the quaternary structure, which is your final protein. This may consist of more than one amino acid chain coming together. And in the graphic to the right, you can see that there is a light gray and a dark gray portion representing 2 different amino acid chains that have come together to make your final structure. The nucleic acids are made up of RNA or DNA. These are nucleotides linked together into a polynucleotide. The term nucleotide is going to mean that you have a ribose or deoxyribose sugar, a phosphate group, and a nitrogenous base. The nitrogenous bases are going to pair complementary. We have 4 different bases in DNA. Adenine, which is represented with A, and thymine, represented with T, bond together, and guanine, represented with G, and cytosine, represented with C, bond together. The DNA is going to form the shape of a double helix. It looks a lot like a ladder with rungs going across that has been twisted. Your DNA is going to end up forming your genes, and it will pass the information from the parents to the offspring of the next generation.